Hey everyone, it's Trent here, and we are here with our second part of our seed oil and heart health conversation, specifically the link between diet and heart health, or what I like to call the cause. And I'll preface this video with saying this is going to be an extensive video, both in terms of length and content. And if this is your first time really diving into the subjects we're going to talk about today, it can seem pretty mind-blowing because there are a lot of physiological, pathological, biochemical, so on and so forth, pathways, mechanisms, explanations that we're going to talk about. And it really is a lot to take in at once. So once again, if this is your first time diving into this subject, I would recommend kind of taking it in chunks. And I myself am actually going to record this video in chunks. So you might see my hair change. I need a haircut. I need to trim my beard. But another thing I wanted to mention is specifically with this subject, always go see your healthcare professional. My word is not the end-all be-all. I've tried to do my very good due diligence in encapsulating what I believe is scientifically profound, the truth. I have resources, I have links, but once again, don't take this as medical advice. Go see your healthcare professional. This is just meant for education. So with that stated, let's go ahead and get started. All right, so a couple changes I'm going to be making with the videos before we get started on the first slide is I'm going to be moving my head around as to not block things like I noticed in a few of the other videos. And with this one, I'm really going to have to do a lot more of just pure slide reading because I don't want to misspeak. And I feel that I've taken a lot of time to put it into these slides so that way I don't go on rambles. I might a little bit, but it's going to be more, boom, boom, boom for this style of video. So what do we mean by heart disease? Heart disease is a broad term that includes atherosclerotic cardiovascular disease, heart failure, arrhythmias, valve disease, and cardiomyopathy. Just some examples. There's more, but those are some. In nutrition, the focus is usually arterial atherosclerosis, and the simple description is chronic gradual plaque formation in arteries supplying the heart. You can get things like heart attacks, brain, you can get things like stroke. In the body, you can get things like PVD or peripheral vascular disease, pain in the limbs, so on and so forth, which upon rupture can form a clot and either block the immediate area, thrombus, or travel elsewhere, embolus. So this is separate from venous-related clotting problems, and this is a little bit of a side note, but I feel it was important because venous-related clotting problems, most importantly or most famously, infamously known, is a deep vein thrombosis or DVT. A DVT is triggered by acute venous stasis, endothelial injury, and hypercoagulability. So a DVT is a clot, which is fibrin-derived, which we'll get into a lot more of this, and you'll be more versed in the language as we go on. So it's a clot formed first by these three factors where red blood cells can become trapped within this clot, eventually building up to either block the venous return in the leg, thrombus, and if a piece breaks off, it can travel as an embolus throughout the venous system, commonly blocking pulmonary arteries. So the biggest difference between what we're going to be talking about in terms of arteries is we have plaque that forms, which then ruptures, which then forms a clot. Whereas venous-related problems is a clot that forms first, which can then rupture. Now, when we talk about plaque that forms into a clot or a clot that forms by itself, that's also called, and that's why I put in parentheses here, a thrombus. When this thing breaks off and travels elsewhere, it's called an embolus. So this can happen either in the arteries or the veins. I feel it's important to know that we can use the same words for two different systems. Some of the pictures I have here are based on the most recent 2024 to 2026 data. Heart disease remains the leading cause of death in the United States, followed by cancer and unintentional injuries or accidents, right? So we're talking about the number one killer of people within the United States and a lot of countries, to be honest, worldwide. Continuing a little bit more so on the right, what we see here is the veins or the vascular system in terms of venous-related clotting problems in the upper right-hand corner. So we see the DVT and how we have gradual clot buildup, which can then break off and go to the lungs for a pulmonary embolism. It's the most infamous example. And on the bottom right, we have atherosclerosis, where we don't have, and this is important in terms of language, we don't have a clot forming first like we do in the venous system. We have plaque building up. And then if we continue to build this plaque up, it can eventually rupture, which then forms a clot. So there's a difference in the arteries and the veins, and we're going to be focusing in on the artery style or the artery context of cardiovascular disease as we continue on. So, arteries are living tissues, and it's important to think of them as dynamic and not just pipes that carry blood because there's a lot of different factors at play within arteries. So, just describing some of the layers, the endothelium or the inner lining of blood vessels, which is what we're going to focus in on. You have an outer layer, very simple description: outer layer, middle muscular layer, and the endothelium inner layer. So, the endothelium or that inner lining of blood vessels regulates vasodilation and constriction. It helps with vascular permeability. It reduces inappropriate clotting. It assists in immune signaling, things like white blood cell movement, so on and so forth. And it produces things like nitric oxide used in a variety of different functions. Healthy endothelial function supports blood flow and vascular integrity. And this is crucial to what we're going to be speaking on soon, is because when we have damaged endothelium and lots of action going on within this and beneath this endothelium layer, we start to have plaque formation. And once we go through this, it'll make even more sense. But once we have the inner layer being destroyed and messed up and plaque is accumulated, we start to incorporate things from the middle layer, and then they can come to proliferate upwards. We'll talk about that more, but just know that this is where we're going to be focusing right now, is that inner layer of these arteries. Okay, so excuse the wardrobe change. I am trying some stuff for a wedding that I have coming up, which is my own wedding. And I didn't want to have to put everything on, take it off, put it on, because I really wanted to talk on some of these points. So I'm like, let's just record. And this is an important slide because when we talk about things like lipoproteins and cholesterol, it would help to know what you're actually speaking on when you utilize some certain terms and get certain blood marker tests done, right? So lipoproteins are transport particles. LDL is a lipoprotein particle carrying cholesterol and triglyceride cargo, where ApoB is the structural protein marker of atherosclerotic particles, right? So, to summarize that, what we have here to the right is an LDL lipoprotein particle because the cargo it carries, those triglycerides and cholesterol, those don't dissolve well in blood, right? So, it's just like if you have water and oil, they don't mix. These proteins allow water and oil to mix in a way because it carries these fat-like particles throughout the blood. So when you get a test done, usually you're like, oh, my LDL cholesterol, my bad cholesterol is high, or whatever it might be, right? Usually tests measure LDL-C, which is a look at how much cholesterol is within these particles. So an LDL-C number is different than the actual number of LDL in your blood. It's just a reference for how much cholesterol are they carrying. Whereas ApoB, which is becoming the new gold standard for heart prevention, disease, so on and so forth, is a protein that's located not only on LDLs, but on many atherosclerotic particles. So lipoproteins that can contain cholesterol, that can be damaging, that's what ApoB measures. Some things like LDL, VLDL, LP(a), things like that. So when you have a measurement of an ApoB, it's more of a direct measurement of cars on the road that could be damaging. That's the typical metaphor that I hear. So it's like ApoB is the amount of cars that you have on a highway, the highway being those arteries that we talked about, right? So that's important to know because although those two can correlate, if LDL-C goes up, typically ApoB goes up because typically you have more cholesterol containing particles, they're not exactly the same thing. So keep that in mind. All right, because cholesterol does not dissolve well in blood, like we talked about, it must be transported inside these lipoproteins. So we already spoke on that. Cholesterol itself is a waxy, fat-like molecule that is essential for human life. It is used for building and maintaining cell membranes, things like helping with fluidity, producing steroid molecules like testosterone, estrogen, and other cholesterol-containing particles, making bile acids for digestion, things like fats, and even serving as precursors such as for vitamin D. So we kind of spoke on this in the first part as well, looking at cholesterol, but it's important to bring up again. Cholesterol is not a toxin or a poison, and it is a required structural molecule used by every cell in the body. Remember those cellular membranes we looked at in part 1. The issue is, in cardiovascular disease, it is usually not cholesterol existing that's the problem. It is how cholesterol is transported inside certain lipoproteins, namely LDL, but those aren't the only ones that can cause damage. How many of those particles circulate, that's a better marker for ApoB, and whether they become retained in the arterial wall, which will make more sense once we go through the cascade of actual plaque buildup, formation, etc. So Let me go ahead and open this link here and we'll speak on these points a little bit more. So what we have here is a 3D model of cholesterol, and I have another thing pulled up we'll talk on in a second, but I think it's important to actually visualize some of these components we're talking about. So what we have here is carbons in gray, hydrogens in white, and we have a hydroxyl, which is an OH group, in red. That's on the third carbon, just the way that it's numbered, right? What's important to know is that although we still have carbon and hydrogen bonds, you can tell that they're differently arranged comparative to the triglycerides we looked at in part 1, right? But both of these components, both triglycerides and cholesterol, don't like to be dispersed in water. So that's what makes them hydrophobic, right? Scared of water, or you can call them lipophilic, which means they love fat in a way, right? What's really interesting is if we turn the structure, rotate it like this, what you see are ring-like structures. 1, 2, 3, 4. And this is the building block of many hormones. So because of the way that this is set up, I'm going to go ahead and move over here. It looks almost identical to testosterone. We have estrogen. But you can tell that the building blocks, the foundation, comes from the cholesterol. So when people say, hey, cholesterol is needed for a lot of steroid and hormone synthesis, that is true. And you can see now it's because of the actual chemical makeup of it. Now, testosterone is a little bit different than estrogen. Estrogen is a little bit different than cholesterol, but they're all within that same realm. They have those ring-like structures. Testosterone here, double bond O, another hydroxyl group here. So technically, if you were to start counting cholesterol, this is the first carbon, the second, the third. So you can tell the third carbon switched out an OH group for a double O group, right? Estrogen in the first ring gets aromatic, so you see these double bonds within this structure, the first ring. So just little differences that showcase that they all kind of come from that foundational structure. And that's why it's important for steroid hormone synthesis, but also using things like membranes, etc. Not going to get too much into the weeds on this topic, but I thought that was nice to see. All right, so now we're really going to get into the meat and potatoes of this conversation, and it is a lot, as you can tell here. It's going to be a lot of slide reading. I'll go on some, mini rants if I need to, add some context. But know that this is not the end-all be-all. Go ahead and, once again, speak to your health professional, look at other sources, whatever you need to do. But I feel that I've done a very decent job in summarizing a very complicated subject. And a lot of this, once you go through everything and you come back, it's like, oh, that's what that means. Or, oh, now I understand how this works in this context. So I'll try not to jump the gun with some of these points and what we'll talk about, but just know that one pass through of this is probably not enough unless you're a super genius. It's looking at it over and over and making those connections for yourself, which I try to make here, but a lot of it has to come from mental connections and mappings. Then it makes sense. So rant done. Let's go ahead and get started. So the dominant evidence-based model showcases that ApoB-containing lipoproteins circulate in blood. These include LDL, VLDL remnants, IDL, and Lp(a). Quick summary. VLDL produced by your liver, it delivers triglycerides to the body. So it's the liver produces it and then says, hey, go deliver these triglycerides. It has some cholesterol, but mainly triglycerides. Once it does its job, it can come back. It can either be reuptook, it's done, or it can turn into IDL. IDL can then turn into LDL. LDL at this point has a lot less triglycerides, a lot more cholesterol, which is why it's typically used as a cholesterol marker, right? LDL-C. Lp(a) is a genetically influenced lipoprotein particle that is directly linked to a lot of the things we're going to talk about. It's separate from these three, right? But once again, it's connected to CVD, and it's typically recommended to get it tested at least once. It's genetically influenced, and diet really does not change it that much. That's why it's recommended to at least get it checked once. So, number 1, ApoB-containing particles circulate in the blood, delivering lipid cargo. 2, entry into the endothelium arterial wall happens for many reasons: damage, metabolic status, etc. But this also occurs naturally regardless of status due to the small size of ApoB-containing particles, meaning when you have these in the blood, regardless of your metabolic status, there's going to be some entering of those of the arterial wall. This is really big because a lot of people argue that, hey, if you have healthy LDL, which we'll talk about later, if you have healthy, large, buoyant LDL, it's not going to enter, it's not going to cause plaque. No, mathematically, the actual size of them, they're going to enter irregardless. Now, metabolic status can make some of these things worse, but just know that it happens irregardless. I'm going to say this 100 times because it's a really big point that people try to argue on. This is a key step, the key step is the retention of these particles within the arterial intima, that inner layer that we looked at, not allowing normal return and initiating a cascade of downstream pathology. So, a summary up to this point is we have these particles in the blood. These particles in the blood, because of their size and because of their location, they can enter that inner lining of the wall. The key step in terms of plaque formation is after they enter, do they stay there? And what happens after they stay there? So that's where we're at right now. It's important to note that even in a metabolically healthy person, some of this happens. Okay? Remember that, please. ApoB-containing particles continuously pass through the endothelium to some degree. Some transient retention occurs, and reactive oxygen species, ROSs, are produced normally by mitochondria and cellular metabolism in the area. Mild oxidation can occur. All of this is irregardless of a metabolic status of a person, meaning by pure mathematics, if you have a large amount of particles over a long enough period of time, some of them are going to enter and some of them are going to be oxidized, irregardless of metabolic status, okay? That is a huge thing. Please remember that. It'll make more sense once you come back and look at all the other stuff, right? Let's go ahead and continue here. All right, I had to move my head here. The big argument and the big question a lot of people usually come down to is LDL or ApoB or is cholesterol causative? Meaning, is it cholesterol's fault that we have plaque formation, the initiation? In a mathematical sense, yes, it is. You need substrate or raw material to enter the wall lining to initiate plaque formation. However, context does matter, and this is not the only impacting factor. How damaged or oxidized are the LDL? Is there chronic low-grade inflammation within the person? Endothelium dysfunction, and so on are all metabolically important, but are downstream from the initial plaque formation. And yes, metabolic status can increase retention rate exponentially. In other words, you need the building blocks, or ApoB-containing particles in this case, to begin to even build. But how much, how fast, how extreme these building blocks accumulate is vastly different from one metabolic status to another. This is a huge point because people tend to argue, hey, if you have healthy LDL, if you're metabolically healthy or whatever, you're not going to build up plaque no matter what. So technically, by that train of thinking, high cholesterol doesn't matter at all because cholesterol is good and is needed in the body. That's pushing one side of the argument as almost too impacting because what you're saying is, hey, cholesterol doesn't matter at all as long as you're metabolically healthy. Other people on the other end, they say, hey, cholesterol is the worst thing in existence, so we need to lower it all the way. There we go, two different sides. We'll tend to see this pattern a lot. But just know that what this is saying is we have particles that have to be there. You can't build anything if you don't have anything to build with. So these have to be there mathematically. Look it up, area under the curve. Area under the curve means if you're at a certain number of cholesterol for a certain period of time, mathematically, some of this is going to stick. It's going to stick and it's going to stay. That's really what it's saying in that arterial lining. However, metabolic status changes the context, and it can make a lot of this worse or better depending upon your metabolic status. So that's a huge point. Come back to that, understand it more with more time, more studying. But if you're just new to this conversation, this is a hugely debated point. All right. And what I'm saying is both matter. Cholesterol number matters. Metabolic status matter. They're not supposed to be taken in a vacuum. All right. It was cool to add at the end. Fun fact, the 9p21.3 locus, which is like a chromosome arm region band, but just isn't part of the genome, part of DNA, right? I think that's an easy way to describe it, is a region on chromosome 9 that's been identified as a key genetic hotspot for increased plaque formation. People of certain variants in this region, especially in European and East Asian populations, have a higher risk of developing atherosclerosis, independent of traditional risk factors like cholesterol or blood pressure. If you carry certain risk variants in this region, it doesn't directly increase your cholesterol, but it creates a more atherosclerotic environment, making plaque formation easier even in the absence of high lipids. So this is something cool. It's basically saying, hey, are you genetically more susceptible to atherosclerosis, right? And you can go ahead and get this tested. If you have like a 23andMe or something similar and you have the raw genome, you can go ahead and like plug it into something to look at this region. But once again, it's in these populations. And basically, you can have variants. You can have one bad variant or two bad variants. So you can either be heterozygous for the bad variant or homozygous for the bad variant. And every time you have one of those, it's like a 25 to 30% increased risk. Now, comparative to smoking, smoking in and of itself can be hundreds of increased risk from baseline, so someone who's normal. But an important thing to mention about this is if you are metabolically healthy and you have lifestyle factors that are anti-plaque formation or anti-heart disease, it can decrease the baseline increase risk of this. So you can really get into the weeds once again, you always can, but with the math. But just know that this is a separate thing that makes the environment more damaging for certain individuals. So there's always more things at play than just diet. But I thought this was cool. You can get it tested, and I'm actually getting mine tested here soon. So something cool. So got a tiny head because there's so much on this, but continuing. We left off with particles that were in the blood. They go into that inner layer of the arterial wall and they're retained, whether they're retained from natural processes or a worsening metabolic status. All right. Once that happens, modification and biological activation of these retained particles can undergo further oxidative modification, aggregation or buildup, and enzymic changes. This makes them much more biologically active. Meaning we have these particles that are in the wall, they're retained, and now they're just becoming a different version of themselves that gives signals for things to come and look at them. That's a very simple way of explaining it, but it's important because the immune response to these stuck and changing particles, especially when modified, like we just talked about, is to send white blood cells, namely monocytes, which turn into macrophages. Macrophage is a certain kind of white blood cell. I like to think of them as very efficient vacuums to go and clear the modified plaque. So we have this plaque that's in the wall, and we have these lipoproteins that are being retained. They're modified, and now it's like, hey, go send a vacuum to go clean it up. All right. Upon engulfing these particles, especially when content includes an accumulating amount of excessive oxidized low-density lipoprotein, or oxidized LDL, these macrophages proliferate into foam cells, causing fatty streak to be seen. This is the earliest visible lesion, right? This is huge. If we look to the right here, foam cell proliferation and transformation of atherosclerosis are primarily driven by the massive uptake of oxidized LDL. And this oxidized LDL can come from diet, it can come from the natural just cellular processes in the area, it can come from metabolic status, right? So it can be oxidized many different ways. That's a big thing to remember. Massive uptake of this oxidized LDL, but this process is fundamentally fueled by a vicious cycle involving both excessive particle number, like we talked about, you have to have the building blocks, and oxidation. Oxidation of the plaque is widely considered a key, often indispensable factor. Macrophages, vascular smooth muscle cells, and dendritic cells take up these oxidized particles rather than native LDL via scavenger receptors, right? Particle number. A high number of LDL particles is a significant risk factor in hypercholesteremic conditions. Excessive LDL particles penetrate the vessel wall, like we talked about, where they become modified and oxidized, right? Many different ways they can be. Small, dense LDL particles are particularly prone to this oxidation. And that brings up the conversation of what is a small, dense LDL particle. That has a lot to do with diet. It has a lot to do with the kinds of fats you eat, which we'll talk about more. But just know, technically not all LDL is considered the same. They all will or can and will enter that wall, but are they going to be oxidized to the same degree? No. Okay? Foam cell formation is not just a one-way street. Once macrophages turn into foam cells, they generate massive amounts of reactive oxygen species and cytokines. Cytokines are like text messages for the immune system, right? They're just calling things, sending things, producing things. There's so many, but just that's what that is. These reactive oxygen species further oxidize nearby LDL particles, leading to more uptake and more foam cell formation. So it's a vicious cycle, right? This is huge. The oxidized portion of LDL is like here, right? So this is your LDL. It goes into the wall, right? Cholesterol-containing lipoprotein particle. It can then be oxidized. Once it's oxidized, it's just weird. It's not the same as it's supposed to be. Macrophage is sent out. It binds to it. It says, I don't know what to do other than blow up, right? Now, if it's normal LDL, and this is a cool thing to know, It can enter the wall and it can leave, and it can also enter the wall. A macrophage binds it, recognizes it because it's normal, and then removes it. But once it's that oxidized weird version, this isn't what it looks like, I'm just trying to say it. Once it's that weird version that just isn't quite what it used to be, macrophage can kind of detect it, doesn't know what to do with it, blows up. Macrophages also do something similar like with tattoo ink. So they're very efficient with things that they know. All right. Fun fact, chlamydia pneumonia, and this is a different kind of chlamydia than what you're thinking of with STDs or STIs. They're both in the chlamydia family, but the STD one is the chlamydia, starts with the T, not even going to try to pronounce it, but just know that we're in the same kind of family, not the same exact bacteria. But chlamydia pneumonia significantly increases the formation of foam cells by triggering macrophages to allow for a much higher LDL uptake, and due to the inflammatory reaction already present, because once again, this is a nasty bacteria, so you're already inflamed. Because you're already inflamed and you're in an environment where it can and will be damaging, they're quicker to oxidize. So this isn't the biggest thing in the world. I just thought it was important to recognize that there are lots of different things that can cause worsening oxidation and foam cell proliferation. But the big take-home point, if you just need to pause this now and think about it, is we have foam cell production, usually via oxidized cholesterol recognition of macrophages, which is a white blood cell. I'm going to stop here. Number 7, these foam cells continue to release inflammatory signals, and when apoptosis, which is cellular death, occurs, a necrotic lipid core continues to form. So we had that oxidized LDL, the macrophage binds, it turns into a foam cell. But once that cell dies, we still have that lipid necrotic core in the center. So that's what that's talking about. The smooth muscle cells, and remember, go back to the picture we talked about. In fact, I will. Smooth muscle cells from this middle layer right here, they migrate inwards towards the endothelium. When they migrate into the intima, or the middle layer of the middle layer goes into the endothelium, it's triggered by those inflammatory signals. This smooth muscle then changes form from contractile, like it's supposed to, right? Vasodilate, vasoconstrict. It changes form from contractile to synthetic, allowing plaque to become a scar-like structure. These smooth muscle cells begin producing collagen, extracellular matrix particles, and elastin-like proteins. All of these work in combination to form a fibrous cap over the lipid core in an attempt to wall off the inflammatory lipid core and stabilize the lesion. This is pretty big here, because what we have are these smooth muscle cells saying, hey, we got all this weird, nasty plaque cholesterol thing here. We don't know what to do. It's dying off. Our immune system isn't working. Let's go into it. It then comes up, and then it doesn't know what to do other than wall it off with a fibrous cap. This fibrous cap, or this fibrin, is the majority of plaque. So now we have this, we had a tiny little fatty streak, right? Once we had oxidized LDL, tiny little plaque, but now we have smooth cells coming up and it's turning it into a big plaque, right? Let's go back. This plaque then continues to grow due to continued ApoB deposition, ongoing oxidative and immune activity, cell death and lipid accumulation, and continued extracellular remodeling. So this cascade is just continuously going in and of itself because there's signals that it sends to increase the beginning parts. It's like a domino effect that continuously reestablishes dominoes that were standing up, and it just keeps going and going and going, right? As the plaque persists over time, ongoing cell death, foam cells and smooth muscle cells, apoptotic body formation, and chronic inflammatory signaling create this gets really complicated, but it creates nucleation sites for calcium phosphate deposition. In parallel, smooth muscle cells undergo osteogenic or bone-like phenotype switching, actively promoting mineralization of the plaque. This results in progressive calcification of the lesion, contributing to the coronary artery calcium score, or CAC. This is why a CAC score is a marker of long-term disease exposure, reflecting an advanced remodeling stage, not early activity. So, as a summary of what that's saying, usually if you're an older individual and you want to look at your risk, do I have plaque built up? Usually you'll look at something like a CAC or something similar that's looking at calcium. You can get like a CT, so on and so forth, and you can get a score back. What that is looking at is the degree of calcification of plaque. But plaque takes some time to calcify. So all that I'm trying to say here is if you get a CAC score that has high calcification, really what you're saying is you have a lot of long-term disease exposure. But what we had here when we had smooth muscle cells proliferating and going into the center and producing collagen in that fibrin, we don't quite have the calcium binding yet. So calcium is more of a late stage degree marker of cardiovascular disease. And if you get it, let's just say a 25-year-old has a lot of this going on, a lot of oxidation, a lot of fatty streaks, a lot of smooth muscle cell proliferation and changing, but they haven't had the calcium yet, their score might come back as safe, right? It might come back as a 0 or whatever it might be, but you still could have plaque forming. I thought that was important too. So just know that calcium is more towards the end of this cascade, and that's where it fits in. Once you have signals for the calcium to bind, and once the smooth muscle cell itself kind of turns into a calcium calling particle. So that smooth, let's go back here. This smooth muscle layer here, once it goes into the middle, it really turns into a transformer of putting fibrous cap here, collagen, elastin, calling in calcium. So this is a really big part of the plaque formation. It's not just cholesterol is built in the middle. Plaque is not the majority cholesterol. It's the majority a reaction to damaged cholesterol being in this area. The majority of plaque is actually this fibrous cap and a lot of these collagen and elastin and extracellular matrix particles. That's what builds the majority of plaque. And then once you have enough time, we have calcium in here as well because it's been called into play. So, big thing, go back, take some time, other sources, but where we are right now is we now have plaque formed, built, and it's been here a minute, and now we have calcium coming into play as well. So that's where we are in our story as of here. What I have here is a pathogenesis of atherosclerosis, a review, and basically I just kind of wanted to put a little bit of emphasis on an article you can go read if you're interested. It's a really good summary, and it kind of helped me summarize some of the points I wanted to look at. It gets into a little bit more depth of a direction we're going to head to, but really what I wanted to showcase was, number one, there are many sources you can find if mine didn't make sense, this being one of them. It talks about risk factors, talks about the proliferation, it talks about some of the things like the thin cap or the thin fibrous cap, right? Like I tried to showcase in some of the images. But if we go down here, this is a really, really good visual summary of basically what we talked about. Lipid retention, we have oxidative modification, and then we have chronic inflammation at susceptible sites, initial fatty streak like we talked about, fibrous plaques, and this is where we are right now in our story, and we'll talk about rupture, thrombosis, stenosis. We'll talk about really honing in next on the rupture aspect. But here's some additional factors in play. LDL particles in blood, it enters, they accumulate, they're modified, they're oxidized, they provoke the inflammatory response like we talked about, they accumulate, we have thickening, we draw macrophages and lymphocytes to the area, take up lipids, they turn into foam cells. This is just a summary of what we talked about. Then we have inflammatory cells, we have the death of them, we have necrotic debris, inflammation, fibrous cap from that smooth muscle cell proliferation, over the lipid-rich necrotic core, fibrous plaque lesions, and then we have a thin cap, and this is where we get to rupture. So this is a good summary of what we talked about and what we will, but once again, if you wanted another source to look into it, they have their own ability to showcase where they got some of their conclusions and references. Just if you're curious or if something didn't make sense, here you go, and they give you things as well. So try to not just hone in only on my own PowerPoint. Okay, another wardrobe change. What we have here is from stable plaque to heart attack rupture. So where we are in the story is we have that mature plaque existing, as we previously stated and went over, with a lipid core, a fibrous cap, which once again has collagen, smooth muscle cells that have went up into that middle layer, calcium deposits, and ongoing inflammatory activity inside. So this plaque, because remember it's a plaque at this point, it's not quite a clot, this plaque becomes vulnerable over time. This is where decade-long increases of inflammation, cap thinning, the lipid necrotic core enlargement, macrophage, which wants to get on those white blood cells, and other inflammatory activity rises. And we have active matrix degradation all occurring simultaneously. So this is why it takes a little bit, right? It takes a minute for this plaque to form, and then for this plaque to weaken, and then eventual rupture, especially depending upon what artery or area that we're talking about. So structural weakening of the cap occurs from macrophage-related enzymes, MMPs, matrix metalloproteinases. Just think these are enzymes and proteins that we have that help break down things, and it's breaking down plaque, which can form a rupture of the plaque. In addition to that, we have degradation of collagen and a decrease in smooth muscle support. So all of these things are happening at the same time, and it's this, once again, continual cascade that eventually breaks down that plaque that we formed. So if we go back to this image, and that's why I like it so much, because we can actually visualize what we're talking about, all of these things are occurring in this plaque area here. We have that fibrous cap, and we're slowly breaking that down until we have a rupture. So a trigger event occurs, often seen in times of blood pressure spikes. Think of like going to super hard, crazy workouts, running, whatever it might be. Physical exertion, circadian variation, morning cortisol surge, etc. So we have a triggering event that just is the straw that broke the camel's back and then breaks that fibrous cap. What we have here on the right side is a cap rupture. So this is a histology slide. So this is an actual area in an artery that they stained so we can see. So of course it isn't blue in real life, but that's the staining method. And what we have here is a rupture. That rupture, if we look at number 5, may occur from the fibrous cap breaking or endothelial layer erosion. That's a whole nother conversation in and of itself. Usually what we're talking about when we mention a clot is plaque rupture, but there is another endothelial layer erosion that's seen in women who have heart attacks very early, so it might not be a decade-long thing. Instead of an actual plaque rupturing, that endothelial layer below erodes, and then the continual cascade, which we'll talk about here, forms. But just for now, just to wrap your head around it, we're just talking about plaque, but know that there's other ways that clot can eventually form. So both of these, once again, lead to the lipid core becoming exposed to the blood. And remember, we have this lipid core that's formed from those white blood cells that are decomposing, that's formed from the cholesterol deposition, and is underneath all of this plaque, because the plaque was just trying to cover it up. So a thrombus or clot forms first by rapidly adhering platelets rushing to the newly exposed plaque rupture. Think of it as once this thing ruptures, there's a bunch of help signals released. The first thing that comes into play are platelets, right? Platelets are, think of it as, the easiest way that I can describe it is your body has platelets to help decrease bleeding and damaged areas, and they typically go to areas to decrease, whether it be exposed tissue, it happens when you get a cut, so on and so forth, and that's why taking a baby aspirin or an aspirin is recommended to decrease the incidence of heart attacks, because aspirin helps decrease the amount of platelets that are formed and that are called into the area. So continuing from here, a blood clot forms over the rupture from a coagulation cascade initiation. So the first things that come into play once that plaque is ruptured here are those platelets. Once those platelets are in place, we have another cascade events, which is its own conversation, once again, called the coagulation cascade. Once you have both these platelets in play and that coagulation cascade in play, you have the formation of a clot. So it is this clot, or at this point it can be called a thrombus, that can immediately occlude or block off an artery where present, or it can break off the immediate area where it becomes an embolus and travel elsewhere to cause a blockage. An example is a carotid artery clot breaking off and occluding the median or middle cerebral artery in brain ischemic stroke. So just think, once we have all of this in play and we had plaque rupture, and now we had platelets coming to the area, then we have a coagulation cascade, now we would have a clot in the area. So it's no longer just plaque. What we would have is something similar to this, not the same exact thing, but we have something, oh, here it is. We have a plaque in the middle, ruptures, and now we have a clot. So those are two separate things. Plaque first, rupture, then clot. That clot can then stay here and occlude an artery, or can travel and then occlude an artery somewhere else. So that's where we are in our story right now. I'll go ahead and open this link up and we can get a deeper look into some of these topics. Okay, so what I have here is what I thought was a very good visual representation of that plaque cascade we just talked about, including the rupture. So what we have here is the shear stressed by increased blood pressure, inflammation, so on and so forth. What we have in this area is plaque, right? So it's not as simple as this. I mean, there it shows macrophages, foam cells, immune cell recruitment, and then we have that smooth muscle, remember, coming from the middle layer up, necrotic core of the lipid deposition products or byproducts, and then we have that fibrous cap. So that was up until this last slide. What I really wanted to focus in on is actually visualizing that blood clot. So here's the plaque, it ruptures, here's the platelets coming in to plug, a thrombus formation. There's also the coagulation cascade in this area. So we have platelets, which then allow the coagulation cascade to form. And then that's really where we get this clot. So there's all that plaque underneath. We have more plaque here, but it's that clot from the initiating events that occludes the artery. So a really big misconception is you just have a lot of this plaque buildup, whether it be cholesterol, whatever people associate it to be. Remember, the majority is actually that fibrous cap. Even in this picture, the majority should be the fibrous cap. And then people think it just builds up and blocks off the artery. That's not what happens, at least in terms of this basic example. What we have is that plaque buildup, and then a triggering event causes the rupture, and then we have all of these cascades, the platelets, the clotting factors, and then we have a clot. And once again, it can either stay here, because at this point we have a thrombus, right? Or it can move, and then we have a embolus, and then it can either block off an area here, or go and continue on and block off another area, whether it be in the brain, or there's certain heart arteries that supply the heart that are typically blocked off. The most infamous is the left coronary artery, or the widowmaker. And it's just very important to know this cascade, because that's really what we're talking about in terms of the triggering event. So once this happens, whether it stays here or goes off and blocks off an artery in the heart, that is a legitimate heart attack. So I thought this was a very good visual representation, kind of puts a face to a name in terms of a metaphor. Oh, real quick, I thought another thing that's important to verbalize is when we have the plaque and we say that there's the fibrin cap right here, fibrous cap, and then all of those events happen and we form the clot, the coagulation cascade, after the platelet plugging. So the platelet plugging forms kind of a tiny little occlusion, and then the coagulation cascade comes in and forms the legitimate clot, the thrombus that we talk about. That also is a majority of fibrin. So you'll hear that word fibrin, fibrous, kind of used in different areas, but just know that that is considered a fibrous cap, which can have fibrin-like materials, as well as fibrin-like materials in the actual clot after that coagulation cascade. So just trying not to confuse people with the similar wording that's used in different contexts. Okay, now we have oxidation and inflammation, or what I like to think of them as are accelerators of disease. So very simply put, oxidation refers to chemical modification caused by reactive oxygen species, or ROSs. We've spoken about this before, specifically in part 1 of this conversation, so just a quick little overview. These reactions can affect lipids, proteins, DNA, etc. And once again, I like to think of them as a game of hot potato where everyone loses, right? And in this case, what I mean is everyone who touches the potato, right, loses. And that's where you have a lot of this damage. That's just a very simple explanation that makes sense in my head. Important markers of oxidation are, if we look to the right here for a little bit deeper explanation, we have oxidized LDL. So this is the typical oxidized LDL is what you hear about in terms of people arguing the metabolic importance of atherosclerosis. We'll talk about that more in depth as we go on. But this is a direct measurement of oxidized particles, levels above 60, yada, 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 right? We have MPO, which is an enzyme released during LDL oxidation. Elevated levels predict heart attack risk independent of traditional markers. That's just saying independent of things like pure particle number or cholesterol number, ApoB. So this is just it doesn't replace ApoB, but it's saying, hey, we also have this thing that we can look at to signify higher risk in certain individuals. We have the 8-OHDG, marker of DNA damage from oxidative stress. People consuming high amounts of seed oil show 50 to 70% higher levels than those eating traditional fats. Now, once again, this isn't the end-all be-all, but just knowing the mechanistic plausibility of seed oil oxidation, which we'll touch more on in part 3. So once you watch all 3 parts, everything kind of clicks into place. But just knowing that there's a mechanism behind seed oils and a certain oxidation product is seen higher in those that eat larger amounts of seed oils. And another one is the F2 isoprostanes, which is a gold standard marker for lipid peroxidation. Studies show direct correlation with dietary PUFA, which is polyunsaturated fatty acids, like we talked about before, intake. So let's go ahead and continue. Inflammation is the immune system's response to stress or injury. Acute inflammation is often protective and healing. You can think of getting cut or an infection. You want the inflammatory response. You can even think of it when you work out. When you work out, you're inducing muscle damage, and you want an inflammatory response to help repair the muscle breakdown and all the byproducts that you're inducing. Whereas chronic low-grade inflammation is damaging. So the word inflammation is really thrown around without a strong knowledge of what you're speaking about. So a lot of times people say, oh, I feel inflamed, or oh, I ate this food and it made me inflamed. And there's not a lot of credibility behind that other than the fact that the person subjectively feels something. So when I like to speak about inflammation, I like to look at it more through an objective lens where you can look at actual important markers, cytokines specifically, of inflammation, things like HS or high sensitivity CRP, C-reactive protein, interleukin 6, interleukin 1, TNF alpha, and you can think of these as chemical text messages that are produced from your body through a variety of factors. It can come from cells, it can come from white blood cells, and it's not always a one-to-one. So like interleukin 6 is known to help increase CRP. So if you have interleukin 6, you're telling your body, and specifically the liver, to increase CRP. But it doesn't only just do that. And then high CRP can go back and enforce other factors. Typically you see interleukin 1 and TNF alpha for like inflammatory response from bacteria, getting white blood cells to where they need to be. So these are just objective markers that you can go into more detail if you're interested. But when you can measure these, now you have an objective lens to look at, are you actually or were you actually inflamed, and can certain things change that? So these are typically tested through blood. Usually you'll see HSCRP as the gold standard for inflammation, but you can get more in depth if you want. So oxidation and inflammation can constantly influence each other, and that's really important. So it's important not to isolate these in separate vacuums, right? So you can have oxidation that induces inflammation. You can have inflammation that induces oxidation. And it can go through back and forth cascades. And when you utilize the language of I was inflamed or I ate this and it caused oxidation, it's very vital to know that it's not A to B to C route. These things are constantly going back and forth on one another. And the more you get into the actual immune system, it was worded to me once that once you try to start studying the immune system, you're given various puzzle pieces and you really can't comprehend the full picture until you have all those pieces. And so in the conversation of seed oils, I'm trying to summarize it down just for very basic markers here, but know that there's a lot to it behind this. But this gives a little bit of context and nuance other than just saying, oh, I was inflamed, or oh, I'm oxidized, or the food might be oxidized. So if we look to the right here, oxidation in terms of ROSs is a biological process where unstable oxygen-derived molecules such as superoxides, hydrogen peroxide, and hydroxyl radicals, those are the 3 big ones. And here's a fun fact, your white blood cells utilize ROSs to kill invaders. So you can phagocyze something or phagocyze something. And so let's just say you have a bacteria and there's certain white blood cells that come and they phagocyze it. And then once it's in the white blood cell, they will actually produce these ROSs as part of the way to help kill the bacteria or whatever it might be in specific white blood cells. So fun little fact, right? Oxidative stress is when ROS production overwhelms antioxidant defenses. It leads to pathological damage, including the lipid peroxidation, membrane degradation, DNA fragmentation, protein alterations. So it depends on what body type and specific area that you're looking at, but oxidation can occur systemically and it can impact a lot of different factors. So just know, from fats to proteins to DNA, it really is damaging. So redox signaling, or controlled oxidation at lower regulated levels, ROSs act as signaling molecules in cell transduction pathways, regulating apoptosis, gene expression, and immune responses. So this is really important, once again, to know that there is normal oxidation that also occurs within your body for specific functions. It's when it goes out of control and you don't have the defenses against it where it can start to become accumulating damage over a longer period of time, especially if you're introducing it not only from your own body's reactions, but from the foods you're eating, so on and so forth. Another example of very oxidizing life choices one can make is smoking. Smoking is known to introduce just so many different particles and cancer-producing elements, as well as a high amount of oxidation, which is part of the reason why it's also linked with higher incidences of heart disease and CVD. So examples of damage and action, we have those lipid peroxidations we've been talking about, proteins, and DNA damage. So this is just a good foothold for one, or if you're interested, to dive into these conversations of oxidation and inflammation. So that way you have a little bit of background knowledge other than just throwing these words out because they sound good. Or with our next slide here, I have inflammation is complicated, because it really is. And I thought this was a good visualization of inflammation. This is in the context of intestinal dysbiosis, so the microbiome's all messed up. But there's a lot of things that we talked about here, and there's some similarities in atherosclerosis, right? We can see that there's constant back and forth between things like C-reactive protein. We have bacterial LPS, so this is a byproduct of bacterial cell wall that can induce inflammation, especially if it's in a leaky gut. Now, I'm not going to go into intestinal dysbiosis for this conversation, but look, we have things, here's the interleukin 6, it can go and influence C-reactive protein. C-reactive protein can go, try to clear things up. This is produced by the liver. If we go in here, we have endothelium damage, so this is kind of in the realm of what we're talking about. Endothelial damage can go down and influence certain transcription factors. It can impair insulin. We have more interleukin 6, more C-reactive protein, more damage. So you kind of get the point. It's not a simple, hey, we're going from point A to B to C, like I mentioned. But when you say, oh, I'm inflamed, understand this is what you're talking about. Now, it might not be this specific intestinal dysbiosis, but the cascade of events, the back and forth, the text messages, the cytokines, the signals, this is really what inflammation looks like, and it's hard to picture it unless you have something that's super complex like this, and it can get even more complex in other diagrams that you can find online. But this is just looking at inflammation so that way you have some context behind some of the things we're talking about, right? Okay, so here we are getting really into the other side of the conversation. So this is typically, other than looking at pure cholesterol number and the cascade that we viewed previously, there's the impact of oxidation and inflammation. So now that we understand how plaques form, how they rupture, and the definitions of oxidation and inflammation, the next question really becomes what determines how quickly this system progresses, right? And this is constantly in debate. Now, one of the main impacts of oxidation, and the truth is it can impact multiple steps of atherosclerosis, but the main impacts, one of them, is the ability to modify lipoproteins, namely retained particles within the arterial wall, producing the previously mentioned oxidized LDL that we just talked about. These oxidized LDL increase the probability of inner wall retention as well as trigger higher immune responses or inflammatory signals due to their recognition as foreign and dangerous particles, causing massive foam cell proliferation, or as we saw before, those fatty streaks, which can then go on to influence the cycle to continue, much like a self-turning wheel. So looking to the right here, in oxidized LDL, both the lipids, including the cholesterol, and the ApoB protein are oxidized, but the critical change leading to atherosclerosis is the modification of the ApoB protein. Studies from the Institute of Health and American Heart Association journals showcase this. Oxidized LDL is formed when polyunsaturated fatty acids and cholesterol within the LDL particle react with reactive oxygen species, or ROSs, producing aldehydes that specifically alter lysine, which is an amino acid, residuals on the ApoB protein. So if we look at the picture here, this is a native LDL. Remember, this is a lipoprotein, and in here we have its cargo. And then, this is a very simple diagram, right? But the oxidation, which can, and this is a big thing, this oxidation can occur via diet or via normal metabolic processes like we talked about before. So now this brings into play what we were speaking on a few slides ago when I said, hey, remember, this can occur naturally. It can. And what we have here is the oxidized LDL, where the lipids and the aldehyde and ketone derivatives are here, and we have that modified ApoB protein, right? Chronic arterial inflammation allows a cascade of increased monocyte to macrophage entry into the arterial wall, increased consumptions of modified particles, in this case we're speaking about the oxidized LDL, and we have a higher foam cell accumulation as well as additional immune cell recruitment. So I'm going to open up these articles because it speaks on it very well. But just as a summary, what this is saying is one, not the only, but one of the main impacts of the oxidation is the formation of these oxidized LDL particles. And when these are in play, it makes retention, the inflammatory signal response, and a multitude of those steps that we talked about increase. Meaning, we still need cholesterol to be present to start the cascade to begin with, but when they are oxidized, which once again can occur via normal metabolic processes or from metabolic dysfunction, which is very highly influenced via diet, once you have these products, we're increasing that plaque formation. So it's a big thing. Let's go ahead and open this up and we can speak on this more. So what we have here is a paper titled Oxidized LDL Associates with CVD by a Vicious Cycle of Atherosclerosis and Inflammation. So a lot of the things we were talking about. This is a systematic review and meta-analysis, which is important to note because when we looked at that criteria of research, we've done it a few times on this channel, this should hold more weight comparative to other studies that were only one intervention or so on and so forth, right? So this is a study of studies, and specifically, I'm going to go down here to the results. They looked at a total of 3 observational studies. So this is a study of observational studies. So there's some credit that can be taken with that, but this isn't direct interventions of a diet. So they're not looking at studies that had people who changed people's diets and then looked at outcomes. They're looking at observational studies. And you can take your time and go through this paper if you'd like. There's a lot to it, so I have to hit some of the hard home run points here. But the biggest one, and they'll repeat this many times, is the results demonstrated that oxidized LDL is significantly increased in participants with CVD in the setting of chronic inflammatory conditions. So this meta-analysis suggests that oxidized LDL may be a useful biomarker in risk stratifying cardiovascular disease, especially in chronically inflamed patients, right? So the cascade that they're looking at is if someone has chronic inflammation, they're most likely going to have oxidized LDL, and this oxidized LDL seems to be associated with cardiovascular disease. So that's the typical route that they're looking at. Like I said, you can go and spend some time here. I've gone through it a few times. I'm just trying to think of the best way to look at specifically what they're saying here, so that way the video isn't longer than it already is. And I think some of the best things is to look at the discussion, right? So they looked at chronic inflammatory conditions, they looked at patients with HIV, rheumatoid arthritis, so on and so forth. What they have here is oxidized LDL is known to have pro-inflammatory and pro-atherogenic properties, and it can predict increased risk of myocardial infarction, right, or heart attack. Additionally, many studies have demonstrated elevated levels of oxidized LDL in chronically inflammatory populations. So these seem to go hand in hand. If you're chronically inflamed, you're most likely going to have higher LDL for the most part, right? I'm going to scroll down here. They are speaking about, while statins are the mainstay lipid-lowering therapy, many people are statin intolerant or cannot achieve goal LDL-C levels on statin therapy alone, and thus require alternative therapy. A lot of times it's statins in combination with other things. There's a very effective but expensive PCSK9, which is an injectable. Once again, please speak to your healthcare provider if you have any questions on any of these medications and pharmacology. I think an important part is, in addition to oxidized LDL as a candidate biomarker for CVD, there's a growing body of literature showing the potential role of anti-oxidized LDL antibodies and other oxidized LDL-related properties, CVD, risk stratification, and promising therapeutic targets. So basically what this paper is saying is, hey, there's enough evidence showcasing that oxidized LDL seems to go up, especially with people who have chronic inflammation. And this could be an alternative route to not replace C or LDL-C or ApoB, but could we add this in addition to those, right? So getting a little bit more specific than just cholesterol, right? So this is the last big take-home point. Other LDL-related lipoproteins, including small dense LDL and Lp(a), which we spoke about before, are prone to oxidation and associated with elevated cardiovascular risk. So this is important right here because some people read this or something similar and run home with this, right? So because of their physical and compositional characteristics, they have a higher affinity for extracellular matrix, reduced binding to LDL receptor, and increased residence time in the circulation compared to large buoyant LDL particles. So what this is saying is that small dense LDL, which more than likely has a higher risk to be oxidized, is more damaging comparative to large buoyant LDLs. So this opens the conversation of saying, hey, not all LDLs are the same. You have a difference, especially in physical makeup. The thing is, all LDL, whether they're large buoyant or they're small dense, can still enter the wall. What this paper is trying to suggest is it's not if you have unlimited amount of LDL large buoyant you're safe, which a lot of people tend to take home with this. They say, hey, if all my LDL are large buoyant, or the majority are, I'm good to go. I don't have metabolic disease, I'm good. What this paper is saying is, although LDL large buoyant are more protected, right, compared to small dense LDL, small dense is more damaging. But it doesn't, basically what I'm trying to say is, you can't just say, because I don't have small dense LDL, I'm not at risk. No, both can and will enter the wall, and then they can be retained and then initiate the cascade. It's just large buoyant LDL have a lower likelihood of doing so, but it goes back to that mathematical probability. You have a large number over a long period of time, some just will be, but not to the degree of small dense LDL, right, and Lp(a), which are also more prone to oxidation. So this is where you can start to see the conversation can get muddled, because someone can read this and take home a point that says, oh, I'm good to go if my LDL are large. They can't be damaged. No, they're just less damaging, meaning they might not induce plaque formation to the same degree as a small dense, but they themselves still can be. So that is a huge, huge point and something I want to drive home, okay? Now, they did note that their meta-analysis had several limitations. The causal association between oxidized LDL and CVD outcomes in our populations of interest could not be defined because of the cohort or cross-sectional nature of the included studies. They're just like, hey, we looked at a lot, but it might not be enough to do a causal claim. We see more correlation, right? So, in conclusion, our systematic review and meta-analysis demonstrate that patients with chronic inflammatory diseases, especially those that they looked at, have significantly higher levels of circulating oxidized LDL as measured by the effect size in relation to increased cardiovascular risk. Thus, and this is what they're really trying to say, oxidized LDL may offer insight into optimizing cardiovascular risk stratification. of chronically inflamed populations or inflamed populations. So they're just saying, hey, this is something you might want to look at, and it seems to have lots of associations, but we don't have the take-home point yet. There's still more research that needs to be put into this. So that's enough for this paper. I got to move on. So what we have here as the second paper is a look at omega-6 vegetable oils or seed oils as a driver of coronary heart disease, the oxidized linoleic acid hypothesis. And I was debating on where to actually put this paper because I could put it here in the second or in the third, and I thought that it would be best if we look at it twice. Number one, through the lens of what we've just spoken about, looking at the oxidized LDL, how it's associated with CVD, how chronic low-grade inflammation can increase it. And now we start to link it into the conversation concerning omega-6 vegetable oils, which we will really go into detail in on part 3. But I thought this was a good segue to look at how the things we've spoken about already can now be turned towards nutrition. So we'll look at this in more depth then. But really what they're speaking about here, once again, is a hypothesis, and I've seen people quote this as evidence, but this is a hypothesis, is the low density or LDL oxidation hypothesis gained traction during the 1980s because it was noted that, in general, native unoxidized LDL does not cause foam cell formation, right? We've spoken about this before. Unoxidized LDL does not cause foam cell formation. We saw that the oxidized LDL really progresses foam cell formation. Now the question is, how do we get oxidized LDL for the most part? In other words, LDL had to become oxidized first in order for atherosclerosis to develop. It was later discovered that oxidized LDL caused direct toxic effects to the cell, recruitment of entry of the monocytes, which we've spoken about, into the subendothelial layer and foam cell formation. So this is the cascade, right? Moreover, oxidized LDL was found to be higher in patients with CAD compared to normal patients, and oxidized LDL was able to better identify patients at an elevated risk of heart disease. So this is kind of speaking about what the last paper was covering. Moreover, oxidized LDL and autoantibodies to oxidized LDL are found in atherosclerotic lesions. So, with all of this, the hypothesis of coronary heart disease concerning oxidized LDL does not get at the root cause. And there's the question again, what causes LDL to become oxidized in the first place? It was later discovered that the oxidation of LDL was initiated by the oxidation of linoleic acid contained within the LDL particles. So what it's saying is, in that lipoprotein, which we know contains fatty acids, they're hypothesizing that linoleic acid, or omega-6s, when they're in the lipoprotein, increases their likelihood of oxidation. Linoleic acid is the most common oxidized fatty acid in LDL. Once it becomes oxidized in the LDL, aldehydes and ketones bind the ApoB, creating LDL that is no longer recognized, like the picture we saw in the PowerPoint, right? But it is recognized by scavenger receptors on macrophages, leading to the classic foam cell formation and atherosclerosis. Hence, the amount of linoleic acid contained in LDL can be seen as the true culprit that initiates the process of oxidized LDL formation, as it is the linoleic acid that is highly susceptible to oxidation. I'm going to pause here, right? People will read this and say, there you go, we got it, don't eat seed oils. We'll speak on this more. I'm going to say that a lot. I've said it a lot already, but this is a plausible mechanism. So just know that what we spoke on before in the prior paper and everything in the PowerPoint can lead way to plausible mechanisms, but the question is, does this showcase itself in other styles of studies, specifically concerning human outcome trials? That's the conversation we'll speak on next. Someone's out there honking, right? So there's the mechanism, right? Let's go ahead and continue. They speak about evidence, right? Greater amounts of linoleic acid oxidation products are found in LDL. Greater amounts of linoleic acid are found within atherosclerotic plaques. A diet higher in oleic, so think of things like olive oil as an easy example. Oleic acid or lower in linoleic acid decrease LDL susceptibility to oxidation, right? Oleic acid is a monounsaturated fat, whereas linoleic acid is a polyunsaturated fat. I'm just going to keep repeating these things until it becomes second nature for everyone. Endothelial cells oxidize LDL, forming linoleic acid hydroperoxides. We've already talked about how endothelial cells, just by their nature, can oxidize LDL in normal metabolic conditions, or if you're metabolically unhealthy and you start to incorporate different dietary means, there's plausible mechanisms to say, hey, we could further induce oxidized LDL beyond what normally already would occur. But what is this, the fourth time now? It normally occurs. So LDL in and of itself can and will oxidize large enough number over a long enough period of time. All right, we're gonna get good at this. We're gonna start second nature, like I said. So meta-analysis of randomized controlled trials in humans found that when saturated fat plus trans fat is replaced with omega-6, high in linoleic acid, there's an increase in all-cause mortality, ischemic heart disease mortality, and cardiovascular mortality. So for this, I'm going to say we're going to click down here, right? And this is the use of dietary linoleic acid for secondary prevention of coronary heart disease, evaluation recovered from the Sydney Diet Heart Study. So what I just said was completely opposite than what we've ever heard, right? So the information that's given in medicine is the opposite of, where's this thing at? The opposite of the control trials, right? So usually you don't hear an increase in all-cause mortality, you usually hear a decrease. And we will specifically speak on this study. This is a cherry-picked study, but we'll talk about that, right? So I'm not going to read all these out, but just know that this is a paper that speaks on very plausible mechanistic means of, hey, diet might increase the amount of oxidized LDL, which we know is pretty highly associated with cardiovascular disease. But is that the only thing, right? So that's enough for now. We'll look at it again through another lens later on, but I thought it was a important thing to bring up, at least where we are now in our story. All right, so I know there's a lot. We're on the tail end here. We're trying to wrap up some loose ends and give a little bit of nuance to the impact of diet and metabolic health, right? Now that we've talked about all these other things, we know that diet directly affects metabolic health, or the body's ability to efficiently process nutrients, regulate energy, and maintain healthy levels of blood sugar, triglycerides, cholesterol, and blood pressure without medication, right? Factors such as ApoB, or LDL, blood pressure, insulin sensitivity, body weight, oxidative environment, inflammatory markers, and endothelium function are all impacted from diet. And I highlighted or asterisked the ApoB-LDL and the inflammatory markers because that's a lot of what we were speaking on previously, right? So diet impacts all of these things. The reason why diet conversations are complex is because of these wide range of changing factors. One food can influence more than one pathway at once. This is a huge thing, right? So like a dietary fat swap is an example, which can muddle the simplicity of recommendations, etc. So one example of metabolic impact is the formation of glycated LDL. This is primarily from high blood sugar, right? So just a fun fact, L-carnosine, which is found in things like red meat, ironically, or you can get it in supplements, is seen to decrease glycated LDL. Glycated LDL, just think of it as LDL that has a sugar attached to it because there was a lot of sugar in the blood. And when you have G-LDL or glycated LDL, you have a higher susceptibility to oxidize LDL. So I won't get too much in the weeds of that, but that's just an example. So this is the big take-home point right here, okay? So for the conversation of metabolic health, which is usually where you find people who don't care about cholesterol gravitating towards, each component of metabolic health can either positively or negatively influence the cascade of plaque formation and rupture that we've already spoken on, Initiation, higher ApoB and triglyceride-rich environments / increased endothelial permeability or damage. So to initiate, to start, to have the building blocks begin to be formed, you need the building block. So there's your ApoB-containing particles. I've said that enough already this video. Progression, right? This is where we see things like chronic inflammation, oxidative stress, insulin resistance, and hyperglycemia impact progression. Plaque stability, hypertension stress on vulnerable arterial walls, extracellular matrix degradation, and reduced nitric oxide signaling. That's where we have things where we can start to rupture more easily. In acute events, so prothrombotic states where you're hypercoagulable, you have a high rupture or clot risks. All of these things are influenced at the same time, which is why it's hard to describe the impact of metabolic health, is because it impacts everything. Every single step that we talked about in terms of plaque formation, acceleration, plaque rupture, so on and so forth, can either positively or negatively be influenced by almost every impacting factor of metabolic health, right? So there's so many different things, and I thought this was the easiest way to word it, is because you can't isolate. Like high blood pressure, it can make plaque stability worse. It can also influence some initiation because you can have higher ApoB go into the arterial wall. This is an example. And you could do that for every single metabolic marker, right? But just know that it's all influencing. So you need the building blocks to get started, but once you have that in place, everything else from there on can be impacted by metabolic health, right? So overall, high blood pressure, insulin resistance, visceral obesity, so the fat around the internal organs in the stomach area, and poor glycemic control don't usually create one isolated problem. That's what I'm trying to say here. They create an internal environment that makes plaque more likely to begin, more likely to grow, more likely to destabilize, and more likely to trigger clotting. This is huge, and this is why people really harp on metabolic health, is because it impacts everything. But you can't just say that metabolic health is all that matters, because once again, we need something to initiate it. So this right here is both conversations, both sides of the story that people try to proclaim. There's people who say that cholesterol is all that matters right here because you need it to initiate. And then there's people that say, well, look, metabolic status impacts everything, right? So this is both conversations in once. This is the middle ground. And this is the same theme that we've seen a million times. Well, we will if we haven't already. In a lot of these conversations is where's the middle ground? And this I thought was a really good way to word it. So yes, metabolic status and metabolic health is vital. It influences everything about plaque formation, right? As well as rupture and the acceleration of all of it. But it's not in isolation, okay? And you can't have any of this to begin with if you don't have the initiating factor right here, okay? All right, so what we have here, continuing on some of these themes, is metabolic health is vital, and I thought I would give a visualization of some of the impacting factors of metabolic disease. Right here, what we have is metabolic-associated fatty liver disease, but we see things that we've spoken on before, things like inflammation and oxidative stress. We have a couple additional factors that we've talked about sometimes with metabolic disease, but just speaking on them again, we have adipose tissue deposition, especially visceral fat, in and around the internal organs. We have insulin resistance, so think of things like leading towards type 2 diabetes in terms of metabolic sense, not type 1. And we have the RAS activation, and this is just a cascade that your body goes through directed to increase blood pressure. So if you have a lot of RAS activation, you can see an increase in blood pressure. And all of these things lead to vascular inflammation. We have the lipid deposition, vascular remodeling, vascular endothelial injury, and hypercoagulability. And all of these impact cardiovascular disease separately as well as together, because when we have this combination of metabolic dysfunction, all of these work on all of those steps that we spoke about in terms of plaque formation and rupture. So this I thought was interesting. Speaking on some of the last slide's points, we have the glycated LDL, like I spoke on a little bit, which is a modified form of the LDL created when sugar molecules bind to it in non-enzymic process, particularly during high blood sugar. So key aspects are glucose or other sugars bind to ApoB, there it is again, proteins and LDL, often forming the AGEs or advanced glycation end products. And we'll speak on this in other topics as well, but looking at AGEs, this is a big important factor concerning carbohydrate consumption, but I don't want to go too much into that right now, but just know that these things do exist, right? Glycation hinders the liver's ability to remove LDL from the blood, causing it to linger longer and increasing its density and damage potential. So this is working on both aspects of plaque formation by pure particle number, as well as its ability to oxidize, so on and so forth. And then oxidative damage. Glycated LDL is more sensitive to oxidation than normal LDL, and it is often found at higher levels in the blood, especially in diabetic patients. So looking at these things together rather than separately, right? So let me go ahead and open this article here. So what we have here is a paper from 2025 titled Metabolic Health and Cardiovascular Disease Across BMI Categories. Some of the findings, once again, you can always look at the methodology and the way they did it, but what they did was looked at 11,499 people, 10,383 were categorized into non-CVD, while 1,116 were categorized into CVD. right? So compared to metabolically healthy individuals, the adjusted odds ratio for CVD among metabolically unhealthy status was 2.07, 95% confidence interval. So basically what they're doing is they're stratifying them by BMI, and they're looking at metabolically unhealthy status. Is it associated, which they said it was, with higher odds of prevalent CVD in overweight or obese individuals? So there's looking at a bunch of different factors and saying, hey, do we see an association, right? So, in this study, metabolically unhealthy status correlates with high CVD risk among overweight and obese individuals, but not in normal weight individuals. Our findings support targeted metabolic health interventions for CVD prevention in overweight and obese groups within the US context. So, this is kind of interesting here because they see that it seems to correlate with overweight and obese individuals, which BMI for overweights, I believe, is 25 to 29.9, and obese is considered over 30. But they did not see this association so much so in normal weight individuals. Normal weight, I think, is like 18.5 to 24.9. All of those are approximates, but right around there. So let's go ahead and go down here because they go really deep into the analysis, right? And the way that they did it. What we get here in terms of the discussion, which is usually where they seem to conclude a lot of the evidence, in this nationally representative cross-sectional study of U.S. adults, we found that metabolically unhealthy status is significantly associated with a higher risk of CVD in individuals who are overweight or obese, but not in those with normal weight, right? To our knowledge, this is the first study to investigate the association between metabolic health and CVD based on BMI stratification. So over the last 3 decades, rates of CVD deaths have significantly declined due to advancement in medication and interventional therapies, but CVD still remains the leading cause of death, disability, and disease burden globally, posing a serious threat to human health and placing considerable strain on public health systems. So, as one of the primary modifiable risk factors, overweight or obesity is particularly prevalent on a global scale, resulting in an epidemic. And they've seen a huge increase in deaths, disabilities, all attributed, well, at least associated with increase in weight. And this is pretty prevalent, especially in Western, more modernized nations. We have a really big problem with excess rather than the problems of the past, which was not enough. So there's a bunch of definitions of metabolic health. We're primarily associated with metabolic syndrome. However, actual definitions are quite diverse. We spoke some of this on some of the prior videos. So while lipid abnormalities are like LDL, cholesterol, triglycerides are components of traditional metabolic syndrome definitions, certain people demonstrated that adding lipid measures did not significantly improve mortality prediction when systolic blood pressure, we have diabetes status, all these other metabolic factors were in play. So basically they're trying to open the conversation to say, hey, yes, we can look at markers like cholesterol, like we've spoken on before, but if we extend our net to some of the metabolic impacting factors or metabolically impacting factors, then we tend to see a little bit more of a correlation with worsening metabolic health and cardiovascular disease. So they're using a new definition of metabolic health, and they found that metabolically unhealthy status was associated with increased CVD mortality compared to metabolically healthy status, right? Via the cascade of events that we talked about, and every single impacting factor of metabolic health can impact every single one of those steps. So if you were to list them all out, it'd be like over 100 different individual things, which is why I try to summarize it to hopefully as understandable or some understandable points. So we're not going to go through all this here, but despite the novel findings of the study based on a representative U.S. population, several limitations must be acknowledged, and this is important, right? So first, the cross-sectional study design prevents the interpretation of the association between metabolically unhealthy status and CVD as indicative of causal relationships. Therefore, prospective cohort studies are necessary to confirm causality, right? So just because something's correlated, it is not causing, right? I mean, that's a big thing. Correlation does not equal causality, right? Second, data on CVD were collected through self-reporting, which may be subject to recall bias. So this is looking at some data where the people actually had to tell the researchers in terms of diagnosis, foods, whatever it might be. So it's not as easily controlled as a randomized control trial. So that's important to know, that they're looking at cohorts rather than actually doing interventions within these studies. But you can still make some conclusions, right? So in conclusions, the finding of our cross-sectional study indicate that metabolically unhealthy status is significantly associated with high prevalence CVDs, especially in those who are overweight and obese, right? So we've gone over that, I think, 3 or 4 times now, but not in normal weight individuals. These results emphasize the importance of targeted metabolic health interventions, things like glucose monitoring, which, by the way, you can do pretty cheaply at home, $80 for like a 2-week glucose monitor. A lot of them you have to get prescribed, but there are companies that allow you to do that at home. Blood pressure control in overweight and obese adults to reduce CVD burden, even in the absence of weight loss. So given the limitations of cross-sectional data, like we talked about, we recommend prospective cohort studies to establish causality, right? So they're seeing correlation, but we just don't quite know if it's causing, or if metabolic status actually causes cardiovascular disease 100%. But we know that there's at least an association. And they also recommend investigations in diverse populations, such as different ethnicities and geographic regions, to assess generalizability, right? And mechanistic research exploring why normal weight, metabolically unhealthy individuals appear resilient to elevated cardiovascular disease risk. So that raises the question, if you are normal weight and you're still metabolically unhealthy, why aren't they seeing these associations comparative to cardiovascular disease risk? And that's where all the interplaying factors of metabolic disease, where they aren't just A to B to C, like I've said a bunch of times already, this is where it starts to muddy the water once again. So it's interesting to note that there is an association if you are overweight or obese, but there seems to be some sort of protective factor if you're not overweight or obese, at least in this study. Now, there are things, you can be normal weight, have high blood pressure, and your risk goes up. You can be normal weight, have diabetes, and your risk factors of cardiovascular disease go up. But in the way that they looked at it, they didn't see it just by pure metabolic status via the markers that they looked at. So I won't go into this too much. I just thought this was interesting to kind of link some of the metabolic status to cardiovascular disease. And this is just one paper of many that does that. With everything, one of the last questions we have is, why do single cause explanations usually fail, right? So heart disease is rarely explained by one pure factor alone. There's an interaction of ApoB burden over time, blood pressure, metabolic health, inflammation, lifestyle habits like smoking, and genetics, and all these interplay with one another. So one of the biggest take-home messages from this PowerPoint is ApoB-containing particle exposure is essential prerequisite for classic atherosclerotic plaque initiation, but particle burden alone does not fully determine the rate of disease progression or clinical events across different individuals. This is where metabolic status plays a vital role. Similar particle levels can produce different outcomes depending on total risk environment. So we're saying that we need this to initiate it, we need it to even start, but how fast it does, how damaging it is, all of these other questions that a lot of people tend to look at seem to be stemming from metabolic health, right? So you need both pieces of this puzzle. You can't just be stuck on one end or the other, where cholesterol is the only thing that matters. Technically, to initiate plaque buildup, yes, but you also can't say that metabolic status is the only thing that matters. Does it matter to look at the rate of progression? Yes. So you need both of these, right? Missing either one is a key player in continued discourse surrounding the subject, and is often where one can find themselves backing into one or two camps rather than extrapolating from both. This is a really big thing, and you will see this over and over and over again, no matter where you look, right? With this, there's a theoretical question. Well, why wouldn't I just get on medications then, statins, resins, etc., to lower ApoB no matter what? Wouldn't this just stop plaque initiation to begin with? Because we know that we need it to start. Now, once again, go speak to your healthcare professional. This is not medical advice. Go speak to them, right? But in a mathematical sense, yes, this is true. But there are always system trade-offs, right? So each medication has downstream impacts on other pathways other than just its primary target. This is another topic entirely, and important information to receive from a healthcare professional. But this is why risk baseline is important in pharmacological modifications, and is most likely what your healthcare professional will do. They'll look at where you are at, they'll look at your risk, family history, so on and so forth, right? And then they'll see, are the trade-offs worth it? Now, each drug can have different trade-offs, right? The most infamous one are statin trade-offs. I won't even get into that, because then you start to go into the realm of should you not. I'm just trying to blanket this as information, but you can look into some of the trade-offs that some people have higher amounts of, some people don't have any, but there are realistic trade-offs. But the question is, are the trade-offs worth the risk? And if the risk is high enough, usually your healthcare professional will say yes, they are, right? So common false binaries in heart disease debates are things like it's all about cholesterol versus all about inflammation, it's all about saturated fat, or it's all about sugar. And if ApoB is normal, then nothing else matters, versus if inflammation is low, lipids don't matter. So these are the two camps that a lot of people often fall into, right? The truth is, multiple pathways can be true at the same time, and they are. I'm going to open up some of these Reddit things like I've done before, just so we can see examples of these conversations that people have. So we're back here on Reddit in the cholesterol subreddit, where someone's asking, does LDL really matter? And no flack to anyone or any of the people that you see here. I'm just using these as informative examples, right? So this person is basically asking, hey, the common consensus is LDL and cholesterol does matter, but there's a lot of people, especially in the carnivore and keto group, that say cholesterol does not matter. It's all about the size of the particles, so the large, fluffy, buoyant versus small, dense that we talked about, ratios, oxidative stress, sugar, etc. And they really just want to know, hey, what's going on here? He's a 24-year-old male, so right around my age, in good shape, and he wants to know because his last blood test showed that he has high LDL of 150 nanograms per deciliter, which is it's an intermediate high number. But his triglycerides are around 70 and my HDL is in the 80s. So, I mean, just looking at a blood panel, you can see that LDL is technically considered high. His triglycerides are considered lower, which would show that he has pretty good metabolic status in terms of the way that his body is hopefully handling fats and sugars, and his HDL is high. Now, the old way of thinking was if your HDL is high, it negates the high LDL, but it tends not to be like that because, once again, large enough amounts of particles over a long enough period of time, technically you could and would have some retention, plaque formation, etc. So here we go. LDL matters. You are likely laying down plaque, right? There's the plague, but so this is automatically in one camp. LDL is all that matters. You got plaque building up. Even though he's 24, metabolically healthy, technically, if he was laying down plaque, that's up to talk to a healthcare professional, right? You'll hear me saying that a lot. So this guy saw a guy in the carnivore sub with an LDL over 500. So they're basically saying that, hey, these guys are going crazy with the cholesterol. They're picking one camp versus the other. How's he not dead? Heart disease can take decades to progress. Carnivore followers believe they are bulletproof from saturated fat. I did for 18 months and then saw the light. So as you can see, people, this is just a great example, I thought. One camp versus the other, right? So it's just, you can just go over and over and over again. You need both low triglycerides and low LDL to be healthy. So what this person is saying is low triglycerides are an example of hopefully being metabolically healthy, and low LDL for low particle burden over time, right? So he doesn't know why the keto head people can't seem to figure out that you should have both, right? So this guy was formerly in the carnivore and keto camp, previously Mediterranean, low dose statin and LDL of about 70. Carnivore diet and no statin, his LDL was close to 200, right? So he's believing the influencers and all the stuff that you see that cholesterol doesn't matter. And then he found groups that say cholesterol does matter, and now he's keeping a low saturated fat and incorporating more high fiber. Hopefully high fiber to help decrease certain amounts of cholesterol because it binds the cholesterol and pushes it out if it's soluble fiber, things like psyllium husk, as well as just overall microbiome health, right? So he started back on the statin. So now he's back on pharmacological interventions, and he will retest soon. There's too much science behind what is advocated here versus the fringe groups and medicine influencers promoting what they consider as real science. So I'm not going to read through all this, but these are just really good examples of people struggling with finding what the truth really is. It's all one thing, it's all the other, and you'll see that people tend to pick a side and they got to stay on it. And speaking on that, let's go ahead and look at some of the other comments that aren't just anti-cholesterol. All right, here we find ourselves in the other camp, and that is the keto subreddit. So this person's asking, has anyone actually checked their cholesterol? This guy's 34 male, he's not really worried, best shape he's ever been in, and he's on keto for 4 years, and he's just kind of asking, hey, has anyone actually checked these things, right? So technically, the keto camp is going to lean one way compared to what we just looked at, right? And I won't diss some of these people here, but looking at this, I thought it was a really good example. Worrying about cholesterol is like worrying about the number of firefighters that you see in your area. They're the good guys, because saying what he's saying basically is cholesterol is sent out to help repair damage, which is true in certain cases. But then he goes on to say what you should be worried about is what's setting off all of those fires, primarily inflammation and insulin resistance caused by excessive continual carb intake. Keto fixes that. So obviously this guy is very deep into the metabolic status is all that matters camp, and he's just saying cholesterol in general. So cholesterol just doesn't matter as long as you're not inflamed, right? What we looked at, hey, what do you even mean by inflamed? And what cholesterol are you talking about? So on and so forth. So let's continue on here. My cholesterol is pretty gnarly and definitely freaked out my doctor, but I believe what matters the most is your triglycerides to LDL ratio. If that is below 2, you should be fine. You just have to trust the process and let keto do its thing. Next, my LDL cholesterol climbed 25% over 2.5 years. HDL dropped about 10%. The standard American lipids panel folk claim I am higher risk now, but my triglycerides happen to fall by 70%. This is a marker, once again, of metabolic status. So it looks like his body is handling things better metabolically, but his LDL is climbing up, right? What I can't decide is if I give a crap about my LDL being at 185, which normal markers, you'll hear people say you want to below 130 for risk, like just normal everyday risk. Some people push below 100, but if you have a high risk like family history of heart attack, or you have some CAC, that scan we talked about that's high enough, once again speak to a healthcare professional, but some healthcare professionals will push to LDL being below even 40 in the newest recommendations that just came out in America in 2026, which reflect the European standards that have been going around just for the last few recent years. So basically what I'm saying is this is considered pretty high in the new standards that just came out, right? And his HDL is being down to 40. I've read so much, and I'm leaning towards, as a standalone health marker, blood cholesterol is very low on the scale of do I really care? It's wild to me that when I get a physical, there's no conversation about purpose, meaning, stress, emotional health, nor is there any stress testing or actual physical tests, VO2 max testing, which is a measure of how well your body can handle oxygen, stressing, balancing, so on and so forth, right? So this is at least in the West in America, right? So this person started keto about 3 months after being prescribed statins. By my next doctor visit, I lost 30 pounds and they saw no need. So perhaps he saw a decrease in cholesterol numbers that the physician was looking at. So some people see better cholesterol numbers within keto. Some people are seeing worse, but those were just examples of people who have heard about a lot of the plausible truths of metabolic status's impact on cardiovascular disease, but are kind of closing their eyes to the other side of the conversation, just like the other people were, vice versa. So just good examples. You can find a million of these all over the place, but I thought this kind of hammered the point home that people actually have these questions and consistently debate them, and they tend to find themselves in one of those two camps. You're missing the other half of the puzzle, and a lot of it is you just feel like you have to pick one or the other. Technically, you don't have to, right? So we're going to wrap up here. All right, so we are finally here, last slide, and I thought it'd be good to end off with the idea that better questions lead to better nutrition debates. So nutrition arguments often fail because people ask low resolution questions. Things like, is this food good or bad? Is nutrient whatever toxic or healthy? What diet is the best one size fits all? What single marker matters most, right? We just saw some of that. Better questions should be context rich. So things like, what food replacements, keyword replacements, can impact my health the most? How much or how often does this certain food impact my health? How do I incorporate my metabolic complex into food choices? What outcome am I looking for, whether it be lower ApoB or weight or whatever it might be, right? How do food choices change in different populations or time frames? Because it's different for every single person. And what are the trade-offs? There's always going to be a trade-off, whether it's medication, food replacement, whatever it might be. What are the downsides? What are the benefits of the choice that you're making? So, with all of this in mind, nutrition is rarely about absolutes. It is usually about context, trade-offs, and better comparisons, which is a huge point to keep in mind because we're going to dive into the topic of seed oils, right? So all of this is just background information, part one, and this is part two for the seed oil conversation. And there's a lot of background, which is why you see I had to separate it. But this is where everything that we've discussed over these last two parts can come into play, specifically with the conversation about are seed oils healthy? Are they negative? Are they positive? What do they do? And what are their impacting factors specifically on heart disease, which is why I did such a long PowerPoint on that topic. So I hope this was not too boring. My best advice is to go back, rewatch some of the complicated parts of this, go look at other sources, have a good fundamental understanding, both of part one, looking at things like dietary fats, definitions, etc., and heart disease, at least to a ability to understand where we're going to lead in terms of seed oils, and then we'll really get into the weeds there. I just finished writing that presentation. I have to record it still, but it is a lot of papers and a lot of words, and I was like, I can't even add a lot of pictures because it just, it didn't make sense to add pictures. So we'll get into that. I'll try to make it as fun as possible, and I really think that once you see where I'm heading with that, it'll make sense. But hope you enjoyed this video. Hope you're going to look forward to the next one like I am, and I will see you guys then. Thank you very much for watching.